Okay, so I think we should get started. It is my great pleasure to introduce our cyber series lecturer today, Dr. Bo Chen. Bo joined us a few years ago. How long has it been, Bo? About 3 years ago now?
3 years ago.
3 years ago from Yale University. Bo is an associate professor in ophthalmology. His laboratories are on Annenberg 22. He also has joint appointments in the departments of neuroscience and cell developmental and regenerative biology, does beautiful work related to retina. So Bo, please take it away. Thank you for joining us.
Yeah, thank you for the introduction. I'm starting share my screen.
That looks good.
All right. It's a great pleasure to share our interesting research on generating new neurons and protecting old neurons in the retina. Our research is to using these two strategies for vision protection and restoration. Retina is a thin layer of neural tissue in the back of the eye, and it has all the layers as the same layers layout and a simplified system in the central nervous system is the outpost of the central nervous system. It has several classes of the neurons, including the sensory neurons, photoreceptor cells. Photoreceptor cells mediate the first step in vision. And it has several types of interneurons. These are relay neurons. They sense the information from photoreceptor cells and they relay the visual information to the output neurons, which is retinal ganglion cells. So ganglion cells send the computed visual information from the retina to the brain. That's the way our vision is formed and perceived. As in the other parts of the central nervous system, retina is subject to various degenerative conditions from genetic defects and environmental factors. So the two types of the retinal neurons are particularly vulnerable. Photoreceptor cells, those are rods and cones, and the retinal ganglion cells. So these types of the retinal neurons, photoreceptor in the outer retina and ganglion cells in the inner retina, they are in the great needs of being protected and replenished. So retinal diseases, a lot of the retinal diseases comparing the vision, they take a stage-wise progression, and they are chronic. Many diseases takes years, even decades to develop, from early signs of vision impairment and to the advanced stages, which can lead to complete blindness. So take an example of the AMD, which is standing for age-related macular degeneration, affecting the central part of the retina that is responsible for visual acuity. So at early sign, you probably you experience just blurry, but as the disease advances, your vision really got compromised, and at the advanced stage, we lose the majority of our vision. So when we think about how to restore vision and slow down the disease progress, we can think about, we can protect these retinal neurons from dying and delay or prevent their death, or we can generate new retinal neurons to replenish lost retinal neurons. So in terms of disease progression from early intermediate, so when the existing neurons, old neurons are still there, we can use neuroprotective strategies to delay the degeneration. However, in the advanced stages, when the majority of neurons, retinal neurons died, there's nothing to protect of. So the regeneration probably is the only way to restore the vision. But ideally, these two strategies are not exclusively against each other. They can overlap. For example, at the early or intermediate stage, if we can protect the old neurons and in the meantime generate new neurons, so we can ideally, we can fully restore vision. So part one of my talk is about the generating new retinal neurons, focusing on generating new photoreceptor cells. In terms of the regenerative medicine in the retina, there are two major strategies to generate new retinal neurons. One is stem cell transplantation. So this strategy involves in vitro coaxing of the either embryonic stem cells or IPL cells into the retinal cells, and then purify the photoreceptor cells and do subretinal injection. And so those transplanted photoreceptor cells, hopefully they can integrate into the retinal circuitry and restore vision. So this type of regenerative strategy, as you can see very obviously here, that the retinal subretinal injection can cause retinal detachment, which is type of injury to the retina. That is by itself is the damage, and also we cannot cover the whole retina. So everything you do the transplantation is patchy. So that is only cover the certain areas of the retina, and also the host of the individuals may produce immune responses to repair the foreign transplants. So another strategy is not involving introducing foreign cells, but using our own stem cells that reside in the retina itself. The question is if we can coax our own retinal stem cells and do the self-repair. Do we have these type of cells? The answer is yes. So we have the type of cells, which is the glial cell type, also known as Müller glial in the retina. Müller glial is the primary glial cell type in the retina. Actually, they share the common progenitor cells during early retinal development. The single progenitor can give rise to both Müller glial and later on to different retinal neuronal cell types. So Müller glial cells, as depicted in blue here, they have both lung processes running up and down across the whole thickness of retina. Like their other counterpart in the brain, glial cells, they provide a array of structural and nutritional support for the neurons because they intertwine and interact with the neighboring neurons. And in the retina, Müller glial actually serve and very interesting another function. They are a pool of potential stem cells in the retina. However, the regenerative capability from Müller glial cells varies dramatically among species, such as in zebrafish, they have the full regenerative capability from Müller glial cells. They can repair the whole retina and restore the full visual function. In mammals, we lost the regenerative capability from Müller glial cells. So other species such as bird and amphibians, they return partial regenerative capability from Müller glial cells. So here shows the section of the retina, how regeneration happens in the zebrafish. Using light damage, very strong light stimulation of the retina. This is why we need to wear sunglasses to protect our retina, the photoreceptor cells. So as you can see, after 96 hours of strong light treatment, all the photoreceptor cells labeled by rhodopsin, which labels rod photoreceptor cells, and blue opsin labels cone photoreceptor cells, they all died. However, miraculously, 3 days after light damage, both rod photoreceptors and cone photoreceptors, they completely fully regenerated. What happens with this regeneration is because of the Müller cells. As you can see here, single Müller cells. Upon cell death, light stimulation causing dying of the photoreceptor cells. Müller cells automatically re-enter cell cycle, enter the first phase of regeneration, proliferation phase. Single Müller cells can produce a cluster of the Müller glial-derived stem cell little clusters, multiple daughter cells produced. And after the this proliferation phase, so the daughter cells they differentiate into photoreceptor cells. So the lost photoreceptor can be replenished, and one daughter cells remain as Müller glial cells. So there's no depletion of the Müller population. And in zebrafish, the system is so beautifully designed and executed. So whenever there's a damage of any type of retinal cells, this the automatic repair mechanism just kicks in and fully restore the visual function. However, as I mentioned, the mammals, including humans, mice, humans, we don't have this regeneration in our retina. But the good news is the regenerative machinery still exists in the mammalian retina. So over many years, a lot of labs, they try to wake up the regenerative capability in mammalian retina, but they have to rely on very severe retinal injury. The most common injury used is AMDA. AMDA, as you may know, is a excitotoxic damage to not only to the retina, to a lot of the regions in the brain. Actually, the AMDA mediated excitotoxicity plays a role in the neurogenesis in both acute and chronic damage in both the retina and in the brain. So as you can see, after AMDA damage, a few, not all Müller cells, a few number of Müller cells re-enter cell cycle labeled by BRDU in green here. But the damage is so obvious, you can see many more cells are labeled by tunnel, which is red signal here. So that many cells labeled by tunnel in red are retinal ganglion cells. So as I mentioned before, retinal ganglion cells are the only output neuron in the retina. They send the long axon projection into the brain. So killing the majority of RGCs seems to be counterproductive for regeneration, as those RGCs cannot be regenerated in the first place. So when we first start doing research on the regeneration, we start by answering the questions. So any of the damage needs to be translated into the cell signaling. So if we understand what the signal is being triggered by the injury, maybe we can have a way to just activate the signal, the signaling of the cells, and thus we can bypass the injury and to activate the regeneration. We look at the many different signaling pathways. Wnt signaling pathway is one of them. Wnt signaling pathway is famous because they play a very important role in maintaining the homeostasis of the stem cells in many different tissues. So in stem cell self-renew, the proliferation and also differentiation. Showing here is this brown signal in here is the stem cell leash embedded at the base of the microvilli in the small intestine. So here shows the canonical Wnt signaling pathway. So there's a lot of different Wnt ligands, but without the Wnt ligands, so the effector protein of Wnt signaling pathway is beta-catenin. So beta-catenin is being associated with destruction complex, and then that destruction complex keeps phosphorylating beta-catenin and leads to ubiquitination and proteasome-mediated degradation of beta-catenin. However, in the presence of different Wnt ligands binding to its receptor furazole and co-receptor LRP, so beta-catenin is released from the destruction complex and then translocated into the nucleus, activate target gene transcription. So we started looking at Wnt signaling by hours after the MD injury. So as you can see, within a relatively period short time, 12 to 24 hours, multiple Wnt ligands being upregulated. In the meantime, when the antagonist DKK and the wave is being downregulated, all indicating the activating of Wnt signaling pathway by the MDA injury. So when we block Wnt signaling using the chemical blocking Wnt signaling, MDA is no longer be able to trigger the proliferative effect of Müller cells. That means Wnt signaling indeed is the injury-induced signaling pathway activation for Müller proliferation, and it's required. So next we ask a question whether we can activate the signaling pathway to activate the Müller cell proliferation without an injury. So we use AAV-mediated gene transfer to deliver beta-catenin to Müller cells. As you can see here, Müller cells are labeled by cyclin D3, and after the gene transfer, Müller cells re-enter cell cycle labeled by EDU. And after the cell cycle reactivation, they move up and down the apical and basal surface. So this is a typical behavior of the proliferation that is very similar to progenitor cells. When they undergoing the proliferation phase, their cell body move up and down. They don't stand still. So next, we really want to know the downstream signaling, the molecular mechanisms of this activation. So when the beta-catenin has a lot of different targets. One of the important targets is the Lin28. Lin28 is an important stem cell factor. The Lin28 is a microRNA binding protein. They bind to microRNA in the nucleus and the cytoplasm. And also, so this binding leads to the inhibition of the maturation of the microRNAs. So we will look at whether Lin28 could be the downstream target of Wnt signaling. So here we look at both the mRNA level, protein level. So after beta-catenin gene transfer, the RNA level of the Lin28A and Lin28B is being upregulated. And also when we look at the protein level, so Lin28 is not, the expression is not detected in the mature retina. So the stem cell factor is quiescent. So after beta-catenin gene transfer into Müller cells, and you can see the protein level of Lin28A, you can see it started being detected in Müller cells. So next we look at the molecular level, whether this downstream activation is at the transcription level, or use the chromatin immunoprecipitation assay. And we identified two prominent binding sites of beta-catenin on the Lin28 promoter. Those two binding sites are relatively close to the start codon ATG. And so in the Lin28 promoter activity assays, as you can see here, the beta-catenin activate the Lin28 promoter, both Lin28A and Lin28B. So after this binding site has been mutated, so beta-catenin no longer be able to activate the Lin28A promoter or Lin28B promoter. So from here, we can see the downstream target activation is at the transcription level and the promoter region. So to see if the Lin28 activation is essential or required for the Wnt-induced proliferation of Müller glial cells, we knock out the double knockout in both Lin28A and Lin28B. After knocking out the Lin28A and Lin28B, beta-catenin no longer be able to activate the proliferation of Müller glial cells. So that means this is a signaling pathway that is very important for the Müller proliferation. If we activate this signaling pathway, we activate the Müller proliferation without causing the injury to the retina. So the next important question, because we're doing neuroscience, we always want to know something about neuron, not just stop at the glial cell fate. So we want to know, are new neurons generated after Müller glial cells being active into the cell cycle by Wnt gene transfer. So in order to know if the neurons been generated or not, so after beta-catenin gene transfer, we dissociated the retina into single cells and apply a panel of the cell type specific markers, neuronal markers, to probe if any of the cells, the cell cycle active Müller cells labeled by EDU, they co-express marker for various types of neurons. So we found and a subset of the cell cycle reactive Müller cells, they actually express marker for PAX6, syntaxin, and N. So these markers are markers for retinal interneurons, such as amicron cells. But when we apply a panel of the markers for photoreceptor cells, none of these cell cycle reactive Müller cells express marker for photoreceptor cells. Because photoreceptors is an important cell type, we want to Müller cells generate new photoreceptor cells. That is the interest, the type of interesting cell type we want to regenerate. So this leads to us to think about we need to probably not able to be sufficient just doing the one-step reprogramming. We probably need to do a second step of programming to further reprogram Müller glial cells to photoreceptor cells. So the first step of we can activate Müller cells into the stem cell status, so they re-enter cell cycle. So then we may need to add gene transfer of other factors to guide the differentiation of the cell cycle reactive Müller cells to become rather photoreceptor cells. To do that, we take advantage of a cocktail of 3 transcriptional factors. So these 3 transcriptional factors, namely OTX2, CRX, and NRL. So these 3 transcriptional factors are essential and playing the very important role during early retinal development. Loss of any of these 3 factors will lead to great sacrifice of production or differentiation or the maintaining of the rod photoreceptor development from retinal progenitor cells. So the way we look at taking a look at the retinal development, because we are thinking regeneration could be happening by following the path of the retinal development when progenitors give rise to different type of retinal neuron, including photoreceptor cells. So the information needs to be translated into the specific transcriptional factors. So using this cocktail of these 3 transcriptional factors for the second gene transfer, we see the Müller glial cells undergoing progressive differentiation into rod photoreceptor cells. When I say progressive, is like over time we see they started to undergoing differentiation rod photoreceptors down the rod photoreceptor cell fate. So as you can see, we can see at least 3 stages. And the initial stage, when we put the rod photoreceptor reporter into Müller glial cells, they turn on the rod photoreceptor reporter in Müller glial cells, so which never happens in the normal Müller cells. That means after second gene transfer, early after gene transfer, some Müller cells started to differentiate into rod photoreceptor cells. So over time and the intermediate stage, which is the most interesting part of the differentiation, as you can see here in panel E, a single Müller cells undergoing asymmetric cell division, producing 2 daughter cells. The top daughter cells is becoming rod photoreceptor cells because they are growing outer inner segments, which is not the feature of Müller glial cells. And then the lower daughter cells is remaining morphological wise, they remaining as Müller glial cells. And they haven't completed the cell division yet because these 2 daughter cells are connected to each other but with two cell bodies and different cell morphology. And then over time, in the terminal stage, the upper daughter cells morphologically becomes very much, if not exactly same, very much similar, look like the native rod photoreceptor cells. Not only they grow outer inner segments, and you can see in the lower, they have enlarged synaptic boudin. So these are synaptic structures they need to in close contact with second order neurons. And then interestingly, the other daughter cells, which is remain as Müller cells. So after they complete the cell division, they become two cells. The other cells turn off the rhodopsin reporter. So that means the other cells is not rod photoreceptors, remain as Müllers. Sorry. So we see this regeneration happens across the retina and some numbers in different quadrants, four different quadrants in the retina. We don't see the regeneration favors a certain part of the retina. Looks like it's pan-retinal. So when we look at the fluorescence image, we're looking at kind of a bigger scale. So we were wondering whether this regenerative new photoreceptor indeed are rod photoreceptors cells. So we need to look at the ultrastructure of this new photoreceptor cells. So the native rod photoreceptor cells, they are very, very so sensitive to the light stimulation. A single rod photoreceptor is sensitive enough to respond to a single photon stimulation. So the reason they are so sensitive is because they have a very specialized cellular structure. So I show a couple. So here is showing on the left, showing they have a very elongated outer inner segment. So the outer inner segment, as you can see in red here, they have stacks of the membrane structures. So these stacks of membrane structures, they are the housing places for the variety of the proteins and enzymes, including rhodopsin, which is the G protein coupled receptor. They respond to light. Single photon stimulation, they can change conformation. So all this light stimulation is being converted to electrochemical reactions within the outer segments. And then the inner segments, so they're just full of mitochondria because they are high sensitive machinery for photo detection. They need a high energy support. They have a high energy demand. And also down lower in the synaptic part, they have a very typical specialized synaptic structure that needs to be connected to second order neurons. So we labeled the newly regenerated neurons with the immunogold and looking at the electron microscopy, the transmission electron microscopy, and side by side with native photoreceptors. In panel Y, you can see in the outer segments region, they have the membrane, stack membrane structure. They look very, very similar to the photoreceptor, native photoreceptor right next to it. And in panel Z, lower, left lower, and you can see the inner segments is full of mitochondria in the new photoreceptor cells. And in panel Z, the upper right, yeah, the Z, you can see the new photoreceptor have the very typical synaptic rod synaptic ribbon structure. So those electron dense, they have just full of vesicles ready to release, and they are in perfect close contact with the second order neurons next to it in the horizontal cells and bipolar cells. So up now, we can see the new photoreceptor generated from Müller cells that are morphologically, even at the ultrastructure level, very similar to native photoreceptors. The question is whether this new photoreceptor integrated into the retinal circuitry and can restore visual function. So here shows the entire visual pathway. The retinal is the outpost, and it sends the light and the visual information from outside world. And all the visual information is being summarized and calculated, summarized into the output neuron retinal ganglion cells. As you can see, these are long tables from the retina. So these are retinal ganglion cells that are sending their axons. They bundle together, known as optic nerve, and to the various different relay centers in the brain, and eventually reach the primary visual cortex, where the visual information being finally processed to support perception. So in order to assess whether the new photoreceptor integrated the circuitry and restore visual function, we look at the output neurons in ganglion cells and the response in primary visual cortex. We did that reprogramming in the mouse model of congenital blindness. These mice are born blind and in their whole life, never been able to see anything. As you can see on the left, the control, these mice have no response in the RGC ganglion cell recordings, either to green light stimulation or UV light stimulation. However, after reprogramming and new photoreceptor routes being generated in these mice, so these mice can respond to light now. So you can see their RGCs respond to green light and to UV light. And they respond better to green light than to UV light, so which is very typical features of native normal rod photoreceptor cells. So we also did collaboratively to record the primary visual cortex by VEP recordings. As you can see in the control, the upper traces, these mice have no response to light stimulation in the primary visual cortex. However, after reprogramming, new photoreceptor generated, so they might start to have the response in the primary visual cortex. However, this response is not exactly the same as wild type, although the response can be as big as wild type response, but not every light stimulation have a response, and the response is delayed. So we done this reprogramming. We provide proof of principle saying that in the mammalian retina, the Müller glial cells can be reprogrammed to generate new photoreceptor cells. But we still have a lot of challenges ahead. So the first challenge is there's still very limited regeneration in mice. So in zebrafish, the regeneration is 4. So whenever even you kill all the photoreceptors, they regenerate, they replenish the loss of photoreceptor cells. However, in mice, there's only one cell division and only one photoreceptor is regenerated. And we don't see multiple is very, very limited regeneration. So the total number of rod photoreceptors generated in our reprogramming procedure is less than 1% of normal number. So that means in the mammalian retina, there must be multiple layers of mechanisms to inhibit the regenerative process going on or ongoing in the Müller glial cells. And secondly, so we are not seeing every Müller cells under the same treatment is undergoing the differentiation into rod photoreceptor. As you can see, the neighboring cells, a few neighboring cells are also being under the same treatment labeled by GFP, and only one of them undergoing regeneration into rod photoreceptor. So we ask this question, whether the Müller cells are could be slightly more or less or not slightly or even significantly different from one another before or even during or after the reprogramming procedure treatment. And so another challenge is, so we've done this reprogramming, although in the adult stage, but in really young adult stage and 1-2 months old mice. So when we want to use the regeneration strategy for restore vision, because a lot of the disease is aging disease, because the people having this disease, they take a chronically long time to develop. And when treatment is needed, it's usually at an older age. So when we do look at this reprogramming, in the first step, the proliferation, you look at 1 month old, you see so many cells being activated. And you at 1 year old is dramatically decreased. The age-dependent decline in Müller activation will be a significant challenge if we want to use the regenerative strategy for vision restoration. And even more challenging is, can we regenerate other type of retinal neuron, including cone photoreceptors and retinal RGCs? So currently, we're doing research targeting these major things, how to improve the reprogramming efficiency to generate more rods and whether we can generate other retinal neurons. So with that, I'm gonna switch gears to talk about the second part of our research. protecting all the retinal neurons, in particular retinal ganglion cells. Most of the majority of the data I'm going to present in this part are unpublished. You probably still remember earlier in my talk. So in order to activate the Müller cells, most often researchers use damage, the AMD excitotoxic damage that can activate some Müller cells. So as you can see, the AMD damage causes the death of retinal ganglion cells, RGCs, and they labeled by tunnel labeled the red cells here. Because RGCs can not be regenerated in the first place. So protecting RGCs is paramount important, because if we can regenerate RGCs, the only way we have for the therapy is to protect RGCs from dying. So protecting RGCs is challenging is because RGCs not only have the cell body residing in the retina, they have very long axon projections. The optic nerve, they project to various parts of the brain. So we need to protect both the cell soma and the axons in order to restore vision. So we're thinking about anything that still needs to be conveyed or translated into signaling. So we've been trying to find what could be the signaling pathways that's been perturbed after the damage. If we can identify these pathways, maybe we can reactivate these pathways and protect the RGCs from dying. So calcium signaling pathway is one of the pathways we've been looking at. I apologize for not being able to release the genes in this pathway as advised by the Sinai IP office as we're going through the patent application. As you can see here in the control retina, this calcium signaling pathway is very much maintained at a high level in the control normal retina. However, after AMD damage, the CAM-K signaling pathway is just in a lot of cells is gone. But you see some cells still maintained, but these cells are not RGCs. These are other cell types, amicron cells, displaced amicron cells in that cell layer. In RGCs, this calcium signaling is really done after the damage. However, when we reactivate these pathways in RGCs by gene transfer, as you can see, a specific gene is being activated. Nearly all RGCs are protected from AMD damage. So this is very robust protection. So how about the axons, long axon projections? So as you can see in the uninjured retina, so A, right? So the panel A. So from the retinal side on the left, and the long axon projection to the right. So those distal projections to the visual centers in the brain. And after AMD damage is lower, so the axons are degenerate, gone, damaged, or axons now cell bodies are gone, axons are gone. But after the reactivating this calcium signaling pathway, all the axons, long distance axons, are also protected. Okay, so that is AMD damage, is the excitotoxic damage. So we're looking at the disease model. So in the retina, there's one disease that particularly affects RGCs. That is glaucoma. Glaucoma, so the primary risk factor for glaucoma is the increased IOP, intraocular pressure. So this increased pressure that can cause a significant stress around the optic nerve, and leading to the degeneration of the RGC axons and RGC cell bodies. So to mimic the disease, we use microbeads injection model, occlusion model in the mice, so in which we inject microbeads into the anterior chamber. So this microbeads going into the corner between cornea and iris, block the aqueous humor flow. So this way, the eye pressure is increased. As you can see in panel C here, so after the beads injection, the eye pressure is maintained at a higher level, 8 weeks after injection. And then we look at RGC survival. So about 2 months after the increased IOP by the beads model, about 35 to 40% of RGCs die. But after the reactivation of calcium signaling with gene transfer, a single gene transfer, the majority of RGC are protected. Okay, so this is a disease model. We're still looking at a different glaucoma disease model. So is there any other model we can use to test this? There is a model which is well established, used by many labs, which is optic nerve crush. So if we crush the RGC axons by using a pair, you can see here, using a pair of forceps to crush the optic nerve, so which causes the axon damage. So this is a very acute damage. The axon will degenerate after crush. Not only axon will degenerate, 2 weeks after crush, more than 80% of RGC cell bodies will die. So this optic nerve crush model is being widely used to study RGC survival and axon regeneration. We also use this model because the manipulation is relatively simple. We can use AAV injection to do gene transfer manipulated signaling part, cell signaling in RGCs. And then we can evaluate whether RGCs survive by counting the RGCs and the flat mount. And we can trace if there is a regeneration of axons by doing CDB tracing. So earlier, in our earlier research in the lab, as you can see, after optical nerve crush, if without any treatment or control treatment, so the axons is stopped there at the crush site and labeled by the asterisk. There is no axon going beyond the crush site. So there is no axon regeneration. And also in panel E, you can see the RGCs, 80% of RGCs die 2 weeks after the optic nerve crush. So earlier we found that if we reactivate AKT signaling, so which is a cell growth signaling pathway, also within the mTOR pathway. So when we activate AKT pathway, we see a significant regeneration passing the crush site, as you can see. So any axons passing the asterisk is a regenerating new axons. And also we see a moderately increased RGC survival after activation of AKT here. So currently our research is looking at whether we can manipulate the calcium signaling pathway alone or together with these other pathways, including AKT pathways and possibly other signaling pathways. Can we protect more RGCs and promote axon regeneration? So far we've been using multiple models, AMDA excitotoxicity models, disease models, glaucoma disease models, and optic nerve crush models to study neural protection of RGCs and axon regeneration. So by the protecting of old neurons and the regenerate new neurons, the ultimate goal of our research is being able to restore vision for a lot of different degenerative conditions in humans. So this is our ultimate goal. I think we still got a long way to go, but this is the ultimate driving force for us to keep pushing and doing this research, because basic science research, we encounter a lot of failures, but we never stop there. We'll always come back strong and with different changes, adjustment based on the original design. So with that, I would like to thank my lab members who have carried out this research. We really work as a team because teamwork is most important thing that can help not individual research, but overall, everybody is very synergistically for all our research. And our fundings from NIH and my Growth Family Foundation, when I was recruited in Sinai, and Pew and some other foundations. Thank you all for listening, and I would be happy to take questions.
Thank you, Bo. Any questions for Bo?
All right.
Perhaps I can start. I mean, the Wnt data were fascinating, really interesting. I was wondering if you'd considered looking, or if you have already looked at other pathways that are known to be related to Wnt, interact with Wnt, and could be other candidate pathways to regulate this process. Things like Notch or hedgehog signaling. Yeah, sense you would think to take a look there also.
Yeah, you ask a very good question, fantastic question. So so when we although our initial work is looking at Wnt, because of the challenges we encounter in terms of improving reprogramming efficiency. So I think Wnt is one of the pathways, but there may not be the only pathway or the important pathways, because not every neural cell is being activated, and even the ones being activated is only one cell division. So we're looking at Notch pathway. We're looking at also another reprogramming factors. So these are ongoing work. We're making some progress. We think these pathways may work, will work even crosstalk with each other. We make some progress on the on these other pathways, but I think we're still evaluating how these pathways regulate either independently, how they talk to each other. As you mentioned, Notch, definitely, yeah.
Any other questions for Bo?
Yeah, Bo, I'll ask a question.
Hi.
Nice talk. My question, well, I'm wondering if you can give us a few more details on the involvement of calcium. So I completely understand the NMDA experiment and the calcium excitotoxicity, but for the RGCs, when you do the nerve crush, where is the calcium coming from, and how important is it that you I mean, is it important that that signal happens at the site of the nerve crush, or is it at the soma?
Yeah, that's another great question. So in terms of the crush, so either crush or AMDA, we haven't really looked at the calcium, the upstream, the calcium. We're looking at a little bit downstream of the effector of that. But although we didn't look at the specific for where the source of calcium from, based on my reading, optic nerve crush. So after optic nerve crush, the calcium, the damaged area forming like a bulb-like structure retraction bulb. So the calcium, the people saying it could come in from internal and external also. So like mitochondria or these organelles, yeah.
But I guess a follow-up is that, do you think that this is when you get the nerve regeneration, is this a local effect? Is it all happening at that site of the injury, or does it require the some transcription or some role for the nucleus?
Yeah. Yes, so this is the axon regeneration. So in the field, many studies from Zhigang He lab. So the translation looks like is a very important step for regeneration, because they need to really crank up the protein building blocks. So either the AKT pathway, mTOR pathway is all, synthesize new proteins, synthesize new proteins. So if you block the new protein synthesis, the axon regeneration will be blocked. So lately, there is also great research done looking at the membrane, the lipids build up. That is also pretty important in the regeneration process. And I think because the regeneration is is kind of currently the RGC axon, because it's a long distance, it takes quite a while. Even with the best manipulation, it takes quite a while to grow that long axons into the brain. So although the translation and lipid synthesis is important, they has to be got into the also transcription level, because the local build-up, there are even some local translation, right? They have message in the, and the areas, local damage area, they make a local translation. But those reserves gonna be used up. You have to have the transcription level. You have to have that to keep the sustained supply for regeneration process. Yeah.
Any other questions for Bo?
Yeah, I have a question. Bo, this is Alon. Can you hear me?
Hi. Yes, I can hear you. Yes.
Yeah, excellent talk, as always. Bo, want to take it just to the clinical research level and ask you, in terms of glaucoma, can you comment of your opinion of why some of the NMDA antagonist studies, I mean, with NMDA being, an important factor here, an instigator or part of the pathway, do you know why the trials on NMDA antagonists failed?
Yeah.
What's your opinion of where the problems are in some of the modalities, some of the
yeah. Right. So that research was kind of controversial, right? So in history, so the NMDA antagonist, because the way I think, because our research actually is quite counterintuitive. It's very different. Actually, it's the opposite to previous report. For example, so the earlier research report that NMDA excitotoxicity actually overactivate in the signaling part. But we found that actually it's opposite. It dampens the signaling. So I think the difference, because in terms of disease, is, so everything happens is pretty local, and it depends on, that's the local concentration of MDA, and the antagonist treatment is whether that antagonist treatment in the broader area, they're all continuous across the whole period of time, whether that works the same way at the single cell RGC level to counter the signaling part. That may be very different from the way we look at the single RGC levels. I mean, we've been asked by the reviewers, because why you see so compromised inhibiting of this pathway instead of the people you think they actually activating. So that's why people use antagonist, right? So this is a hypothesis behind this. But when we compare our results with the previous results, is because the previous all look at global level, and you look at the whole retinal homogeny that coming from all the cells. So we're just looking at RGCs and single cell level, and because of the way we look at and the resolution we look at, so maybe there's the increased activation of the signaling in other cells, but specifically in RGCs is actually inhibition. So that's why, I mean, this turns a plus and minus opposite way, right? So then we went further to test. If this inhibition, we kick up the activation, can we protect RGC? The answer is yes. So that actually confirms our finding. So if the previous finding using an antagonist, we're thinking maybe and the single cell level, if you want to only target, not using antagonists to treat the whole retina, just targeting the specific cells we want to protect. So maybe that's where differences. Maybe we just activate the pathway within RGC. So generally speaking, I think in terms of therapy, always drug therapy like injection or eye drops, they're targeting everything. So everything, all the cells being exposed to the treatment is experiencing this treatment. And our way is just look at the target cells, only treat the target cell, manipulate target cells. So yeah, that's a great insightful question.
Yeah, thank you, very helpful, thank you.
Okay, it's a few minutes before the hour, so I want to thank you both for a great talk and thank everyone for joining us. And I look forward to the next seminar in the series. Thanks, Bo.